Hi, everyone. So welcome to this first session of this scientific event of the Manutech-SLEIGHT Graduate School. It's my great pleasure to introduce Hugues as our first invited speaker. Hugues is a research director with INRIA Lyon. He's the head of one of the 20 or 19 INRIA project teams within the INRIA Lyon Center. So he's the head of the so-called AI Strauss site, which is a team that develops numerical approaches for neuropharmacology. Hugues published tons of papers at the interface of molecular biology, but also computer science, AI, and applied mathematics. So today Hugues will talk about simulation and how to characterize how molecules move in living cells. So I hope you will enjoy this talk, and it's time for me to leave the floor to Hugues.
Thank you very much. Thank you, Mark, for the very nice introduction. So it's a pleasure for me to be here today. So I understood that the audience was a bit diverse in terms of origins, in terms of scientific origins. So I've tried as best as I could to give you something that is not focused especially in one direction, but don't hesitate to interrupt me if you think that I will go a bit through a bit of theory for notes. If it's too difficult for you, don't hesitate to tell me. And of course, don't hesitate to interrupt me if you have questions. You're more than welcome to ask questions. I will start my timer because, so I've understood that the goal is to a 2-hour talk, and we'll have a short break in the middle, if it's okay for you. Okay, so to start, I will share with you a memory of when I was a kid, which was some years ago, a couple of years ago. And when I was a kid, I used to imagine how it would be like to be, like an alien coming for the first time to Earth, right? In my great flying saucer. And so you arrive on the Earth, and by night, you fly over a huge city, like, I don't know, New York, Los Angeles, without the fire. And look at that, it's very complex. And my question was, how could this alien, how could he or she understand the organization of the city and understand how human beings are organized in cities? So what you see lights going in directions, going from there to there. You see structure like this, where you also have lights, but the lights here, they don't move. And you see that you have plenty of movement, right? But you suppose that behind this movement, you have some kind of human organization. That's how the city is organized. But how can you infer how the city is organized from this movement, right? That must be very difficult if you're an alien, if you don't know anything about human behavior. And in fact, it turns out that it's exactly the same. That's my picture here. The same feeling I had the first time I've seen a cell, the inside of a cell in a microscope, a modern microscope. Because if you look, it looks like that. That's a single cell. And you see that inside the cell, everything moves. You see there's the stuff that moves there, many movements there. Here you have structure that move a bit, but a bit less, and so on. And it's very complicated. And it's even more complicated that if you have several cells, then you have movements inside the individual cells, but you also have moving cells themselves. It's complicated, right? So it's a bit, for me, it's a bit the same kind of question. How, as a cell biologist, whatever, can you understand, infer the functioning of the cell or the cells on the basis of the movement? So it's a bit like Cell City. It's like maybe the oldest of you know SimCity. It was a video game well, anyway, and it's a bit the same. You have something that moves movements, and how can you infer the organization of the city on the basis of those movements? And of course, it's like that because a cell is not a bag full of water, right? It's full of water, but also inside the cell, you have huge internal compartments. So that's that compartments that are made of internal membranes and that are isolated from the rest of the cell, right? So you have, for instance, the nucleus there, here. Sorry. The nucleus is that's where the chromosomes are, and it's separated by a membrane, the nuclear membrane, from the rest of the cells. You also have, for instance, those kind of compartments that are called mitochondria, and they are producing of course, the energy of the cell. And also you have this weird stuff that is between tubules and pancakes-like structure that is the endoplasmic reticulum, and also those guys there that is weird, like science fiction stuff, that is the Golgi apparatus, right? So all these, they are like internal structure that are very similar to, in the cell city, as buildings or factories, right? That's a bit the same that what you have in a real city. So of course, because you have buildings like that, you have to move things between, you have to deliver things. And in fact, that's why you have movements inside the cell. The same way that you have movement inside a city, because you have location where people live and other locations where people work, and you have to move in between. It turned out that in a cell, you have two major types of movements, right? Maybe I can click on the. No, I can't. Oh, okay, it works now. And the third type of movement is very similar to what you have when you order a pizza. It's like something like a Deliveroo or Uber Eats. Deliveroo is mostly European, I think. Well, it's only that when you have to move things from one building to the other, and for that, the thing, how do you do? You package your proteins or your thing inside the vesicle. So it's like the bag, you know, those deliver guys that have a bag in their bike. You do the same with your proteins that you want to address, and you package them into those. So these are the green stuff. That are the Deliveroo packages, if you will, the vehicles. And you see that the vesicles, they move along those lines, those cables that are called microtubules. And of course, it's a very, you see this guy, and you have a small, it's not a bike, it's a small stuff that walks, that is called dynein or kinesin anyway, and it moves straight ahead, you know? Like, so that's a movie that's not experimental stuff, it's artistic view, but that's how it happens, really. So you have those Deliveroo guys that move from one place to the cell to the other with vesicles that are moving straight ahead into tubules. And how did it use for? You see, this is, for instance, this is an experimental measurements. So you see in white, the inside, this is a neuron cell, and you see in white the transportations of the vesicle that goes from there to there. It's again very similar to what you could see in the street of a city, right? So that's one first Deliveroo type of movement between compartments. I won't focus on that today, and I will rather focus on the other type of mobility. So if you are in a city, you can move with, I don't know, metro, so public transportation, but the other way is also to walk, right? And of course, the other type of walk is walking inside the city. And inside the cells, most of the molecules, they also walk. It's not like, except that their walk is random. It's like if they were like drunk, right? So it's drunk walk or random walks. This is, so for instance, you have here a real experiments, and I will come back to how can we do that experimentally. So what you see there, this is the inside of a living cell nucleus where the chromosomes are. And you have much noise there, but this white spot that you see is a single molecule. Here it's a cymic protein. It's a transcription factor anyway, but you can follow the trajectory of a single molecule, here a single protein, inside a single nucleus of single living cells. And when you reconstruct the trajectory, in the green stuff, you see that it's exactly like my funny guy who was a bit drunk. You see the movement is random, and you don't see really the direct displacement. You have the same thing here, but this time you have several individual trajectories, and that's for a receptor, the EGF receptor on the plasma membrane. And it's not easy to see, but again, you see random movement, so random walking. Of course, you are all familiar with probabilities and statistics. It's not because it's random that it's always the same. Randomness comes with distributions, and distribution comes with parameters. And of course, you can have different motion, different parameters. For instance, you have an example here. So it's a sodium channel on the movement of a sodium channel on a membrane of a neuron. And you see that you have different behaviors. Some of the channels, they have in green, they have random motion that have a very long territory, and the blue and the red just rather say where they were, right? They don't move far away. It's still random, but it's not the same type of behavior. So how come that it's like that, right? How come that you have this random motion, that's drunken molecule movement in the cell? In fact, we know that, how come that it's, I will come back to that, since 2025, something like, since 1925, so around the century, it's because of thermal agitation, right? At the spatial scale that we are talking there, you see in blue, black, most of what you see are water molecules, right? What you have locally in the cell, it's water molecules. And those water molecules, because of thermal agitation, they move, right? They go. Of course, when they meet, it's just like a billiard. They collide and they change their trajectory. So it's a pure deterministic, it's a elastic or almost elastic track. So nothing funny in that, except that it's all deterministic. But if you have so many water molecules, at some point, if you look at a given water molecule, the direction of its displacements and the amplitude of its velocity, you can describe it as a random variable. And in fact, it's in terms of scientific concept, it's a hugely powerful concept. That means that you can have a system that is purely deterministic, no randomness, whatever. But because you have a huge number of degrees of freedom, so you have a huge dimension, then it's so complicated that at some point, it's still deterministic, but you can describe it very efficiently with a random process. So it's false randomness if you want, but still it works. And so all of our water guys, at some point, they have a direction that is random. But of course, your protein that is there, the yellow guy, will collide also with those blue water molecules. And if each time a black water molecule collides with the protein, it will move it to a small direction, right? A small move to a direction. But because the direction of water molecule that collides is random, so the movement of the protein is random as well, right? So the randomness of the displacement of the molecule comes from the randomness of the direction, the distribution of the direction of the water molecules. No. It's random variable. It's distributed over the whole volume. So as a result, what you have is that at each time point, your protein molecule, so here it's a virus molecule, has a small displacement at a random direction and with a random velocity. That's exactly the random walk that we were talking before, right? So we'll come back to that a bit later to see how we can model that, right? But actually, at least that's the source of random walking in the cells. So, of course, the question as a scientist, the question that we have is, well, what are the properties of this random walking in the cell, and what are the parameters? Of course, again, it's random, so it comes with parameters that characterize the randomness. But in living cell, what are the properties or the parameters of these motions? It turns out that it's only since 10 years, something like that, that we are able to reply to answer these questions, and I will show you why. Is it constant? I mean, the parameters and the properties of this random walk. Is it constant? Is it the same for the given cell? Is it the same for the same molecule and so on? The distribution, does it change depending on the size of the molecule? Does the large molecule move fast or slower than a small molecule? Does it depend on the compartments, and so on, so on, so on? So many questions of this type. And of course, if you are a biologist, now you will ask, this is more physicist-like questions, but if you are a biologist, you will ask, well, does it impact the cell operation, right? If, let's say that I change the random walks of my molecules because I change the temperature, for instance, does it affect the operation of the cell? And this means the operation of the cell, they are based on biochemical interaction, biochemical reaction. So does it change biochemical reaction if you change the random walks of the molecule? And of course it should be, because remember that to occur a biochemical reaction needs the reactants to collide. It's not, you have no action, you have no chemical reaction by action at distance. So the reactants, they must meet and collide for the reaction to occur. And of course, the probability that they encounter depend on how they move, right? So of course, if you change this random walk, you will change the way the biochemical reactions occur. But how? That's the main question, right? So that's exactly this type of question that we would like to answer. And that will make the outline of my talk. So first, a bit of theory again. I've tried to be as simple as possible, so I think it will be okay. But if you have difficulties to follow, just don't hesitate to tell me. But we'll see a bit the theory of that is behind. We will talk quite much about the available experimental approaches to see that, because of course, it's if you want to quantify those random walks, you have to measure them in a way or another. So you have to have experimental ways to see that. And it turns out that we have two main experimental ways to see that. The first one is a very recent one, is a single molecule tracking super resolution microscopy. I will explain a bit how it works. But I mean, most of you are coming from optics, so maybe that will be very simplistic. My presentation will be too simplistic for you, but and another one that I really love is fluorescence correlation spectroscopy. It's an old experiment, but it's a very nice one, so I will talk to you about that. And then I will show you at the end how can we exploit those experimental measurements and those theories to answer the question that I've presented at the beginning. That is model selection and calibration, and I will show you how it's easy to do that, or it's easier to do that with machine learning and approaches, right? Okay, so a bit of theory now. So yeah, so how can we model and say something theoretically about this random walks? So as always, I really like to have some kind of historical overview. And in fact, they started at the beginning of the middle of the 19th century of all this story, with the first with the fixed law for the concentration. We will come back to that in a moment. And then the real milestone will be in 1905, the paper by Einstein on that, and we will come back. You will see, we'll see on that. A bit, I won't say much about Langevin's approach, because he's a French guy, so who cares about French guys? And the major updates has been done at the in the second half of the 20th century with those continuous time random walk, fractional motion, random walk on a fractal. So we will go over quickly over that. Okay, so what we are talking about is a theory about how would this random walks occur, which is another name for diffusion. And it turned out that in the theory, there's a real milestone that is at the beginning of the 20th century. Because before the 20th century, everybody was thinking in terms of continuum fluid dynamics. And let's say that rho of x and t is the concentration of your fluid, so the concentration of your protein at position x and time t, right? So the thing you can do is just to say, okay, I can impose a conservation constraint, conservation of the mass. And this conservation equation there, it's very simple. It tells you that if the concentration at x varies during t, it's just that because it's the difference between what came, the flux that came from the left and the flux that left for the right, right? So the variation, the conservation of the mass tells you that the variation in time of rho here at x, it's just the gradient between the things that came from here and the things that get to here, right? It's just nothing is created, nothing is lost, everything is conserved, right? Now the question is, how can we express this flux? And the first one to express was Fichte, who proposed in 1855 that the flux would just be the gradient of concentration, right? So the flux is proportional to the difference between the concentration between the positions, right? So if you plug that derivative into this derivative, you'll have two first-order derivatives. So that's a second-order derivative, and you see here that the time derivative of the concentration is proportional with a d proportional constant to the second derivative of rho, right? Of course, what you have here is what we call the heat equation or the diffusion equation, and that's the first term that was established by Fichte. Of course, if you are in 3D or in several dimensions, this is a Laplacian operator that you have instead, but it's the same idea, right? And the important stuff is that if now you consider that your space is infinite, you have infinite space, and you consider that initially all the concentration is located at x equals 0, you can compute easily the solution of this differential equation. It's what mathematicians call the fundamental solution. Physicists will call it the propagator. And this, of course, the solution of that is that rho is varies as a Gaussian function of time, right? So the solution of this equation is that the concentration at time x and at space x and time t is just a Gaussian which variance increases with time, right? So you have this variance that increases, and at infinite time it goes to 0, of course, because you have infinite space. So you can say, right? So that was what was available before the 20th century. And then suddenly everything changed around 1910, right? Because what emerged then, you remember 1910, 19something was the time where Boltzmann started to work on the molecular or kinetic theory for gases, right? That was a point where physicists started to realize that there's nothing as a fluid. A fluid doesn't exist. What exists are individual molecules that collide, and because they collide, the way they collide, that makes what we believe at a macroscopic scale being a fluid, right? And so they started to think about diffusion as something that is related, just as I said before, to collision and to molecules. And the first to do that experimentally, the first experimental observation for that was made by a botanist, a Scottish botanist called Robert Brown. And he just looked at the movement of pollen grains under a microscope. So it was not very deep, but because it was the first one, this type of motion are usually called as Brownian motion because it comes from Brown. But the experimental, the first really precise experimental measurement of that was done in 1909 by a French physicist called Jean-Baptiste Perrin. And he didn't look at pollen grains, he looked at putty, so that putty is mastic in French. And at the time it was nice because you could control easily the radius of the putty grains. And this is an example of an extract of his paper. And he really looked at trajectory of the grains, and with a grid, he could really quantify the motion, how much the grains move, and so on and so on and so on. This was 1909, and in fact, the idea, Perrin was not interested at all into random walks. He didn't care much. He was interested in how can we determine experimentally and precisely the Avogadro number. And I'll show you in a moment how you can go from that to that. And in fact, this paper, because of this paper, I got the Nobel Prize, Perrin, in 1929 or 26, something like that. So we come back to that. But that's, in terms of experimental, it was the first experimental observation of and quantification of this kind of random walks. Now, in terms of theory, of course, the big milestone was made by Einstein in his 1905 paper. So really, I encourage you to read this paper, or maybe not the original one because it's in German, so you have to speak German, and all German because it's 1905. But you can find on the web English translation. It's a very, one part is about Avogadro number, maybe it's less interesting, but what we are going to see there, oh, the equations are a bit messed up, but it's, in terms of mathematics, it's very simple, really, again. So the model of Einstein is, okay, it's a discrete time random walk movement. So what Einstein poses as an hypothesis as my random walker has a movement where it moves every tau, tau time units. And when it moves, when it jumps, it remains when it was. It remains static where it was for tau time units, and then it jumps, and the jump time is infinitely fast. So you jump infinitely fast, you remember where you are for tau time units, and you jump, and so on and so on, right? So discrete time random walk. Of course, that's where all the theory is wrong, and we will see that the theory is wrong because of that, in fact, because a random walker doesn't wait for tau. Between two jumps, it also moves. And in fact, you will have to wait for 3 years for Langevin to correct a bit that. But at least it makes the calculation very simplistic, very simple. So the only other hypothesis that Einstein does is that at each time my random walker walks, right, the jump distance is random. The distance I jump, let's say it's in one dimension, it's random. So it's a random number with a random probability distribution function, lambda. And the only hypothesis he does is that lambda must be symmetrical, meaning that the probability to jump of distance r to the left is the same as to jump the same distance but to the right. So lambda of r is equals to lambda of minus r, right? So that's the only hypothesis. And of course, that's an important one because that means that your distribution is symmetric, whatever it is, and that means that its mean is 0. Of course, that's the same thing, yeah? Right? That means so the mean of your distribution, the average, the expectation of the distribution is 0. That's also true for all the orders moments. The third order moment will be 0, the fifth order moment will be 0, and so on, right? It's single. And then Einstein does something that is, maybe it doesn't look very interesting, but it's very, in terms of physics, he writes the first master equation ever. Of course, he doesn't call it a master equation in the paper because this kind of vocabulary was not available at the time, but that's what it does. So what he says is, okay, let's look at the probability that my particle x, so I have a single walker, my particle is at x at time t plus tau. Well, how can I write it? It's the probability that it was at x minus r, and it jumps from a distance r between t and t plus tau. But it could be also that it was at position x plus r, and it jumped from a distance minus r between t and t plus tau. And so on. It could also be that it was at distance x plus 2r, and it jumped from a distance, and so on and so on. So if you sum all that, and you go to the continuum limit, what you get is an integral. So your probability is integral that over all the jump possible r, that's the probability that your molecule what has x plus r at mt, and it jumped a distance r between both, right? That's very simple. Now what you can do with that, you can do p of x plus r and t, and just do a Taylor expansion. So you know, Taylor expansion is something that you see maybe at the first year in college, something like that. And that's what he does in the paper. So you expand his p of x plus r is p of x plus r times the first derivative of x plus r squared divided by 2 times the second derivative of p, and so on, plus higher order terms, right? And now, if you distribute the lambda r, you have 3 terms. You can put the p of x outside of the integral, because the integral is over r, and there's no r in p of x. So p of x exits, and you have integrals with terms now that are just r and lambda r. So this term is just the integral of lambda of r, and by definition, lambda is a PDF, is a probability description function. So its integral is 1. So this disappears. You remain with these terms. Here, for the second term, what you see r times lambda, of course, is the first order moments, is the average, the mean. And of course, because of this, we said that it was 0. So this term disappears. And the next term that you have is the integral of r squared times lambda. This is of course the second order moment, so the variance. So at the end of the day, what you have is p of x at t plus tau is p of x plus sigma, so the variance divided by 2, the half variance, times the second derivative of p. And then, of course, if you take your tau and you push it to 0, so you take the 2 tau tends to 0 limit, then what you see here, you will have p of x minus p of t plus tau minus p of tau of t. Of course, that's the time derivative of p, right? So at the limit of infinitesimal tau, you get back this equation. The time derivative of p is d times the second order derivative of p in space. And of course, it's the same equation than before. It's the same equation the same equation as Fick, except now that what we have here, the variable is no more a concentration, it's a probability, right? So that means that if you can solve this equation, you can have an expression of the probability of x. Again, this was 1905. If it were since then we had statistical physics. So what would you do now? You will write some kind of conservation. You take the Fokker-Planck equation and infer the probability with the Fokker-Planck equation. At the time, Fokker was maybe not dead, but he didn't write the Fokker-Planck equation yet. So that means that, again, if you take the idea that your space is infinite, then you can solve that. And what you get is that P is just the solution of that, which is a Gaussian distribution. And that's really important because that tells you that the successive position of your random walker, it's just a random variable with Gaussian distribution with mean x0. X0 will be the initial position, and variance that is 2 times d times t, right? And here d is given by the equation there. It's the variance, the half variance divided by the time step. Very nice. So that means that in another way, x of t, I can write the position of my molecule X at time t as the initial position, plus this term that is the square root of 2 dt times a random number that is just Gaussian with zero mean and unit variance. And that's very useful if you are a computer scientist, because if you know how to generate random number with normal distribution quickly, then you can generate quickly this kind of, very simply, this kind of random motion. You just have to generate a random number, multiply it by the square root of 2 dt, and you have your x of t. It's very simple. And actually, that's exactly what you do, and it's called Brownian dynamics, right? That's how you generate a random walk on a computer. You say that the position of x, the walker at t plus delta t, is the previous position times this term multiplied by the random number. Of course, if you are in 3D, what you do one random walk in each dimension, and it's EIID, all these random numbers are independent, and it's very easy to show. Now, another quantity that will be very important is the average. So with this bracket notation, I will use it for the population average. So that means that for me, the average of x of t with bracket notation is the population average, meaning that if you have n molecules, it's the 1 over n times the sum of the position of xi. So if you do that, the mean displacement, the mean of x is 0. That means that so you have all this blue and red and colored dots are random walkers that walk according to this. And what you see here is the mean of their displacement. So in fact, if you look at the mean displacement, it's 0. So the thing goes wider and wider, but globally, the mean displacement is always 0. So a random walker doesn't move on average. So that means that usually people look at the mean square displacement, so the square, the second moment, and that will be important. If you do the computation, you see that the mean square displacement is 2 times d, the diffusion coefficient, times the time. Does that mean that in Brownian motion, the mean square displacement is linear with time? That's really a hallmark of Brownian motion. And that's what you see here. Here, I showed in black the mean square displacement of all those guys there, and you see that it aligns perfectly with a line that has 2 times t, times d dot t as an equation. Well,
assume that your space is not bound.
Yeah.
Is this property still hold?
No. Yeah, sure. If your space is bounded, then this mean square displacement with plateau at some point, and you can use this kind of, but it's a bit more complicated. Right. So again, all my point was to write this equation. Of course, this is 1D. If you have small d dimensions, in fact, I won't go into too much detail, but it's the same. The mean square displacement is 2 times d, small d, which is the dimension of the space, times capital D, which is the diffusion coefficient, times time. And so that's the main goal or the main result maybe of Einstein's 1905 paper. The other one is that in the same paper, Einstein determines that the coefficient, the diffusion coefficient, this half variance divided by tau, is also RT, R is the constant of perfect gas, divided by 6p eta is the viscosity, A is the radius of the walker, and the Avogadro number. So that means that, and that's what the goal of Einstein, in fact, and it will be the goal of Perrin, is that if you determine precisely D experimentally, and if you determine precisely eta, the viscosity, and the radius of your random walker, you can determine precisely the Avogadro number. And in fact, that was what Perrin used with his Petit. He did a very precise measurement of D and A and the viscosity, which is the viscosity of water. And with that, he could estimate the value of the Avogadro number, and that's why he got Nobel Prize 20 years afterwards. Just if you read the paper, so the paper by Perrin is in French, so it's easier than for French-speaking people than Einstein. You will see that Hugues, the estimate he gets, Perrin, is around 7 times 10 to the power 23. So it was the most precise measurement at the time. Of course, as you know now, the constant that we have is rather close to 6.02. So it's not that precise. It was not that precise, but still, it was the most precise variable. Right. So that's important. Really, again, the fact that in a Brownian motion, the mean square displacement is linear with time. So now we can get back to the cell, and you can just measure the mean square displacement of your protein cells. And what happens is that quite frequently, I will go back to the table, very frequently, you don't find the fact that the mean square displacement is not proportional to time. Usually what you see, you have some form of probability, but the mean square displacement is proportional to some power of time, alpha, so time to the power alpha, with alpha that is lower than 1. Of course, if alpha equals 1 is linear, so you get back Brownian motion. In fact, when you also we'll go back to the methods, whatever you have, you can find alphas for different cells like bacteria, yeasts, mammal cells, and so on. Different compartments. C is C for the cytosol, so the inside of the cell, N is for the nucleus, M is for the membrane. So different, but in every case, at every time, the time scales are different, but for all those results, alpha is lower than 1. And the proteins are very different. It's like mRNA particles, transcription factor, receptors. And every time you see random motion, you see some kind of probability between the MSD and time, but it's not Brownian motion. Sheets. That means that all those guys got it wrong, and maybe Einstein was wrong. Oh my God. Well, so what happens? How come that it doesn't work like Einstein predicted it? But in fact, it's just that the cells are much more complex than the model. Initially, the model is a bit that was used by Einstein is a bit like a perfect gas model, right? It's a single molecule. We know other molecules, only water, right? This never happens in cell. What you have is you have in cells, you have a huge crowding, a huge local density of molecules. You have, it's more like inside of a cell is more like a syrup, like it's very thick, very viscous. And this is, of course, those are simulation, but you see that the molecular crowding, this is the inside of a parasite, this is the inside of a synapse of a neuron. And every time you see it's very crowded. There are many, it's not like random walking in the desert. It's more like random walking in a dense crowd. What you have when you go to the soccer play, and when you exit the Geoffroy Guichard. It took me time. When you exit the Geoffroy Guichard Stadium after the soccer play, I mean, you have a huge, I mean, maybe no more those days, but before, 20 years before, you used to have a huge crowd, and walking over the squad is much more difficult and different than to walk alone. So you have collision, but not only with water molecules, you also have collision with other guys like you, other biomolecules, other proteins. So it's not a perfect gas. And also, you have a huge density of biomolecules, which means that you have a huge macromolecular crowding. Again, it's a crowd. So it's not a water solvent. It's much far away, farther away than a water solvent. And because you have crowding, you have all those intracellular membrane and this weird Golgi apparatus geometry, you have very complex geometries. It's just not that like a 3D pure water. And also, because you have molecules, you have interactions. For instance, you have electrostatic interaction. You can have van der Waals interaction. And because of this interaction, your random walker, sometimes it's stopped, it's trapped. And because it's wrapped, this is not just a random walk. So that's because of that the cells are so complex in terms of medium that one needs more complex models. Except that it's not that easy as that, because it turned out that when you start in terms of, if you are a theoretician and you start with the idea of a random motion, that at each time step you move randomly, you will see that whatever you do, whatever you modify from the Einstein model, you end up with a Brownian motion. In terms of statistical physics, people say that the Brownian motion is a very strong universality class, meaning that whatever the small changing that you do to your microscopic model, your macroscopic model, most of the time will be Brownian motion. There are just maybe 2 or 3 or 4 models that can end up with something that is not a Brownian motion, and that we will see now. Let me look at the. So the very first complexification was made by 2 very famous physicists in 1965 by Montreux and Weiss. And what they did is just say, okay, I won't go with Langevin approach. I will keep on with Einstein's approach, except that now the jump, the jump time is not Dirac distributed. It's not tau at each time. So the jump times now, it's a random time tau with some distribution phi of tau, right? That means that, again, the same model as Einstein before, except that now when I jump, I remain when I am for a time tau that is taken from a random sample for the random distribution phi. But that's the only difference. Again, behind it's the Einstein's model. And now, of course, well, the master equation is a bit something more complex because you have to integrate over the jump distance, but also over the resonance times. But again, it's mostly the same type of equation. Now, to solve it, you have to go a bit through like always, a bit through Laplace and Fourier transform and so on, but it's really easy to do. I mean, it's not that complicated. And what you get is the following. Is that if the mean value of tau exists, meaning that if the mean converges, right? For instance, if phi of tau is Poisson distributed, right? You get Brownian motion, so you get back your diffusion equation for the probability of x, and you get back the fact that your mean square displacement is linear in time. So that's again Brownian motion. Things changes when you say that what happens if the mean value of tau, so the mean is the integral of tau phi of tau d tau, if this diverges, so you don't have any mean anymore, and if it diverges, for instance, if it's phi of tau is power law, so it's of tau, then I don't want into too much detail, but in this case, what you get is that the mean square displacement is indeed proportional to time with some power law alpha. So in this case, when your the first moment of tau diverges, in this case, you can have something that looks like what you have experimentally. What does it mean? It means that it's a motion, it's a random motion, except that each time you jump, you remain at the same position for a moment tau, and this, because it's power law distributed, you can have very long stay. So you have a random walk where sometimes you can stay at the same position for very, very long amount of time. That's the difference between exponentially distributed and power law distributed. And in this case, you can deviate for random motion. And now if you look at the diffusion equation, you also almost have a diffusion equation for P, except that you have here, I won't go into too much detail, but this fractional operator that is called the Riemann-Liouville operator. So you have a fractional differentiation, a fractional diffusion, but still, it looks like it's a bit more complex, but it looks like diffusion equation. So this works, right? Again, if I have random stays, but sometimes I can have very long stays, very long residence stays, then I can have this kind of stuff, right? The other one, the other model that we will talk a bit was proposed a bit after Riemann and Liouville, again by a French physicist. It's Mandelbrot, even though, yeah, he worked in the US for most of his career, and that's fractional Brownian motion. So again, that's the same as before. You take the Brownian motion, right? Except that in the Brownian motion, your variance, your number here, your random number, they are randomly distributed with no memory, right? So that means that you have no correlation between the different numbers. What Mandelbrot introduced was a random walk where you introduce memory between the random walks. That means, to make it short, that means that you have a random walks when if at the previous time you went to the right, then you have a tendency to get back to go to the right as well at the next jump, right? So you introduce a memory in the jump distribution. So how does it compare to biology? So it could be a memory or it could be anti-memory, meaning that if you jump to the left at some time, it's more likely that you jump back to the right at some time, at the next time. That's exactly what you have if you have, for instance, an obstacle. When you have a membrane, you bump through the membrane, so you get back to the first direction. So in a way, this fractional Brownian motion describes what happens if the intracellular media is viscoelastic. So you have many like polymers that moves and moves you back and forth because you have viscoelasticity. And of course, with the fractional Brownian motion, I want to go through the math, but you get back your mean square displacement as being time to the power alpha. So you get back alpha less than 1. So again, this is more similar to what you expect in biology. And the final one is random walk on fractal. So this appeared maybe 20 years after, so at the end of the 20th century. And this is related to the emergence of fractal structure. So if you don't know, do you have an idea of what is a fractal object? So a fractal object is an object that is which dimension is not 2 or 3, it's not integer, it's in between. You have objects that have a real dimension that are 2.65, for instance. So how can you measure a dimension when you're a physicist? Very simple. You take a box, a circle. Let's say that, for instance, your object, it's the blue lattice there. And if I have a red point, is that I removed this element of the lattice from the object. Now, how can I compute the dimension of this object? I take, for instance, a circle there, and that has some radius r, and I compute the number of elements of the blue elements that are located within R. And I increase the value of R, and I count the number of elements, right, that are within the circle at distance R. And it turned out that in terms of this usually is supposed to give you a power law, and the exponent of the power law gives you the real dimension. So to be more concrete, again, let's say that you have a lattice, so a square lattice, that is your object. And then you will come and remove some sides of the lattice at random, uniformly at random. So this is what happens, for instance, when you remove 5%, 5% of the sides at random. This is what happens if you remove 10% at random, and so on and so on. So we remove these objects at random. There is a critical dimension, a critical probability of removal there, where the remaining object here, the blue ones, becomes a fractal. That means that the number of blue sides, they are a power law of R, but with some non-integer exponent, right? And this changes everything. So let's say that, for instance, your molecule, your random walker is restrained and must remain on your blue stuff. What happens to the random walkers? How can you describe this random walks? This has been made very famous by a Degen paper, which is, he called this an ant in the labyrinth. So you have an ant, the ant moves at random, but it moves on a labyrinth that is not a homogeneous space. And it turned out that when you are on your fractal object, you can show that's what Havlin and Ben Avram showed, that the mean square displacement again is proportional to t to the power of alpha. And again, of course, that talks much, it says much for biologists, because you can expect that the inside of the cell is so complex in terms of geometry that probably you could have something that looks like a fractal object inside. So you could expect, if the inside of the cell is a fractal object, fractal geometry, you can expect that the mean square displacement indeed scales at t to the power of alpha, right? So again, that's the four main models that we have to explain divergence from Brownian motion. So I will go again to it. Random walk and fractals. That makes anomalous, what we call anomalous diffusion, meaning that non-Brownian diffusion. Fractional Brownian motion, again, that makes non-Brownian diffusion. And continuous time random walks when the jump times are power law distributed. Currently, in 2025 now, that's the only three models that we know that diverges from Brownian motion. Not exactly since 1 or 2 days, well, anyway, but most of them are right? So of course, what we would like to do, we would like to go to measurement, all those measurements that we have here, that I showed you before, all those measurements, and come back and say, what is the model behind that? How come that we have alpha that is less than 1? Is it because it's not Brownian motion, but is it continuous time random walk? Is it random walk on fractal? And that's in terms of Bayesian statistics, that's what we call model selection. And exactly what we would like to do in the coming hour, just after the break. So after the break, I will show you how experimentally we can access to this data. You will see there are very, very wonderful experiments like super resolution microscopy. And how can we do this model selection? Meaning once we have the trajectories, how can we decide what is the model behind? And we'll do that after the coffee break. Thank you.
C'est 15 minutes? C'est 15 minutes? Okay, so we have a 15 minutes break. Before leaving, I would like to ask some of you to remain quieter during the talk, unless if it's for asking questions during the first part. It was pretty unpleasant. There was background noise, so keeping this in mind for the second part after the break. Thank you very much.
Très bien. Non, pas de
Il y en a 3-4 qui parlent, c'est un peu chiant.
Ah, j'ai même pas fait gaffe. Non. Ah non, d'habitude. Oui, oui. Ah non, mais ouais.
Moi, j'aurais envie de dire.
Oui. Et encore, moi, je trouve qu'ils ne font pas de bruit par rapport à d'habitude, mais ils font plus de bruit. Hein.
Ça, je n'ai pas eu. Ça va. Il y a rien qui désagréable qui m'a fait mal.
Merci.
Je peux pas dire merci. Alors, en plus, avec la douleur, je m'énerve facilement. Non, mais oui. Non, en fait, je suis presque venu sur quelque chose qui est très tôt. Ah, merci.
Alors, moi, le moins de 40 ans, c'est pas.
Et soyez quand même un petit peu sympa avec notre conférencier. Mais tu sais, je dis souvent, la jeunesse est une maladie dont on se soigne très vite. Tu vois ce moment? C'est où? C'est à...
Okay, so maybe ouais, je crois. No? Can you hear me? No? Yeah, maybe I'm going to start again. If you have questions about so the first part was a bit more theoretical in a way. If you have a question about that, don't hesitate to ask them now. You can ask them at the end of the talk as well, of course. But if you have questions, don't hesitate. No problem. So now we will jump to a part that is more related to the experimental part and how to analyze the experimental data, of course, with machine learning tools. So again, the question is, you remember that Perrin was doing in 1909 with his putty grains, doing the quantification of the motion of the grains of putty. I would like to do the same in my cell, right? Look at the molecules but it turns out that up to 2010 or something like that, it was just impossible. I mean, I was trained as a biochemist, and then when I was biochemistry student, people used to say, okay, biochemistry is a science where you never see anything. You have to infer what the molecule does through something, right? Through a red marker or something like that. All this changed in 2010 something, thanks to those three guys, Eric Betzig, Stefan Hell, and William Murner, and they got the Nobel Prize for that, right? In 2014, 10 years ago. And the Nobel Prize was for the discovery of single molecule tracking, or at least super resolution microscopy. And I will go quickly on how they did. And it's a technique, a microscopy technique, that allows you to image single proteins, one protein, in living cells. So not dead cells, but living cells. So how do you do that? In fact, of course, it's always the same. Once you know how to do, it's easy to understand. The idea is that how do you do to your problem that you are limited by the diffraction limit of optical microscopy. Yeah, with optical microscopy, the diffraction limit is around 200 nanometers in cell. That means that if you have two molecules that are close to each other by less than 200 nanometers, you won't be able to distinguish them. Typically, you have an example here. You have a single fluorophore there, and what you see is the blur. This is 300 nanometers, and you see you don't see the single molecule. Now, if you image this ring of molecules, you see the same, right? So you cannot distinguish this is one fluorophore with the other molecules. So how do you do for that? The idea is that if you were able to image a single molecule and not its neighbors, you will be able to just very precisely define its location because you do super resolution and you do the opposite, that this is a Gaussian, and you take the center of the Gaussian. So if you manage to image one cell, one protein after the other, it's easy. But how do you do that? And how they did that, they found fluorescence dyes that undergo between on and off in terms of fluorescence stochastically. Right? That means that sometimes if you put your fluorescence dye you attach it to your protein that you want to look at, then sometimes some of the proteins will go up, you will have a fluorescence signal, and down. But because it's stochastic, it's very unlikely that two neighbors will go up together. So if they don't go up together, if the light emit a photon stochastically, well, if you do your experiment well, there is a high chance that your neighbors won't send the photon at the same time as you do. And so that's it. You have a single molecule experiment. And it works very well. For instance, this is what you had. So again, you have those three rings that you want to image. Of course, if you have a normal classical resolution spectroscopy, you see nothing. But if you do this kind of super resolution microscopy, you see that every time this guy first started to emit the photon, then this guy started to emit the photon, and so on. And when you reconstruct all the photon that you have, you see that it's not perfect, but you see the rings that you had initially, right? So that's the whole idea, is to have this kind of stochastic emission of photon. That's how it works. And that means that now you can have a resolution that goes up to 10 nanometer. So now you're able to see with optical microscopy things that are 10 nanometer apart from each other. In X and Y, it's a bit larger in Z, but in X and Y, it's 10 nanometer. So it works very nicely. So this gives you microscopy for single protein, again. And then if you do it like with at regular times, then you have images when you see position there and there and there, and then after some time position there, and then after 20 milliseconds position there. And you just have to track to reconstruct the position of each molecule and say, okay, here at 40 milliseconds, the protein that was here was the same one that was the one that was here. So I have a track between there. And there at 60 milliseconds, it moves a bit. So this protein that was here at 60 milliseconds was this one at 40 milliseconds. So you do just a tracking like you do in any video microscopy or video stuff when you track position between two time frames. And that's how you reconstruct single molecule trajectories. And again, this changed everything in the 2010 or something. So that's a single molecule tracking super resolution. The pros and cons. Of course, what you can see individual molecules. That's very nice because again, that's like Perrin with his putty. That's very nice. You have a direct representation of the trajectories. You don't have to infer the trajectories. Just you see them. It's there. But there's cons as well. The first that you have to do the tracking. So you have to reconstruct and say, okay, my protein that I see here at this time frame, it was the one that was there before. And this kind of tracking is full of reconstruction error because the time, of course, the time span between two imaging, if it's too long, it's very difficult to be sure that if you see a protein here, it was the one that was here before, or another one that went there, and so on and so on. So it's very difficult to do the tracking without error, right? And then, of course, they're crossing each other and so on. So that's a bit of pity. And you have a low temporal resolution. It's very difficult even nowadays, to look at trajectories that are less than 10 milliseconds apart, right? So maybe 10 milliseconds it looks small for you, but for biology, 10 milliseconds is really long. And usually, and very quite often, and I will go back to that, this is limited to short trajectories, because in fact, what you do is that you image a plan or a slab, right? So a thick plan. But if your molecule comes, it stays in your imaging plan for some time. But if it goes fast, it moves away, and then you lose him. So that means that if your molecule diffuses fast, then usually you have very short trajectories. You cannot have trajectories that have, I don't know, 200 positions in a row. And usually that's what happens, for instance, in the nucleus. Turns out that in the nucleus, usually the protein, they go fast and they move very fast away. So it's very difficult to do this kind of stuff in the nucleus. It's much easier in the membrane, in the plasma membrane, so the external membrane. Usually you can do it easier, much easier. And then the experiment itself, it's still difficult. I mean, if you are a naive biologist, you know how to do confocal microscopy, right? But if you want to do this kind of palm or storm experiments, you have commercial microscope, but it's still something that is for experts. So there is an alternative, and it's a very old one. And it's called fluorescence correlation spectroscopy, that it dates back to the 1970s, something like that. And it's a very nice experiment. Really, I love it. It's very simple. Let's say that this is your sample there. This is what you want to image, and your molecules that move are the white dots there. And you just put the light on the sample, right? You light it. And so, of course, when some of your molecules go inside the illuminated volume, if they are fluorescent, if you tag them with a fluorescent tag, they will emit photons, right? That's the principle of fluorescence. And what you do, you just record the amount, the number of photons that is collected by your photon detector, right? So your primitive measurement is a photon count. It's the number of photons emitted by your molecules per time unit. This is very simple to do. You just have to modify your protein of interest by putting a fluorescent tag on it, and that's it. Now, what does it see? I mean, of course, let's say that you have a very simple motion. So you have a blue one that goes ballistically, so straight ahead, no random motion, but just to illustrate, the blue one is very slow, like 100 micromolar per second, and the red one is very fast. Of course, the blue one, it will start to enter the illuminated volume, so the number of photons emitted will increase, and then when it exits the zone, it will decrease. So you have a very depth, a very large emission time, right? Now the red one, because it goes very fastly, very quickly in the zone, you have peaks, the same peaks, but their width is quite smaller. So that means that if you look at the width of what happens there, you can say this goes fast, this goes slow, and this goes fast. Yeah. There? Yeah. Oh, yeah. This is just an illustration. Yeah, sorry. Yeah, it should be periodic, of course. Yeah. You know, this is just an illustration because, of course, you never have this kind of stuff. But at least that means that if you look at the autocorrelation of the signal, so how the signal is similar to itself, of course, the autocorrelation will be much larger when it goes slow, and it will decrease much faster when the molecule goes fast, right? And that's all what you do. You just say that you take the number of photons, you autocorrelate the signal with itself, and you measure how the autocorrelation decreases with the delay of the autocorrelation delay. And the general rule of thumb is that if you move the autocorrelation to the right, the larger delay, you have slower diffusion, and if you decrease the amplitude of the autocorrelation, you have higher concentration. We will see that in a moment, right? So it's for me, it's a nice experiment. How it works. So for instance, for it to work correctly, you have to have three hypotheses, right? The first one is that your medium, your volume is isotropic, homogeneous, so there's no special direction and so on, and it's infinite. The second one is that your motion is Brownian. So you have Brownian motion, that's why you have your heat equation. There, C is the concentration of molecule at position R and time T. And the third is that you have to explicitly say what is the illumination, meaning that when you put the laser in your cell, how will the light intensity be spread, the PSF if you want, how it will be spread into the sample. For instance, let's say that you have a Gaussian illumination, so you have that is larger in Z and smaller in X and Y, right? And if you do that, you could very simply compute the number of photons that are emitted by time units. It's just C of R times the intensity in R with this contribution of Q, which is the proportionality, which is some kind of cross section if you want. So you integrate that, and then what you get at the end, I won't go, it's a simple calculation, but I won't do it, is this kind of exact autocorrelation. The autocorrelation G of 2 is 1 over N. N bar is an average number of molecules in your sample, divided by 1 over 1 plus 4D2 over this constant omega xy and omega z, right? So that's why that the computation, that the exact value of the autocorrelation, that means that if you measure x of tau experimentally, you can fit it with this expression. It's non-linear fit, it's easy to do. The amount, the maximum value of G of tau gives you the inverse of the average number of molecules that you have in your sample, right? On average, because it's diffusion, so it goes on in and out. And if you look after a decrease of G of tau by a factor of 1 over 2, or over 2 square root of 2, then you have this time tau d there, and tau d is just 1 over d, basically. And so if you measure tau d, you measure d exactly. That's simple. Yeah. Yes, or you are in a dilute regime, so you don't have the plus. It's supposed to be, I mean, so it's photon emission. So the only thing that could happen is that when one molecule emits a photon, another one will be close by and will absorb it. If you are dilute enough, you don't, because again, what happens there is that if your N is too large, then this will go down, and so you won't see your decay of the autocorrelation. So this works only if N bar is small enough. So you have to be in the dilute regime, in very dilute range for it to work. Yeah. Okay, so pros and cons as well. So for it to work, you have to add the number of molecules in your imaging volume to be small enough. Small means 3 or 4. So that means that you can look at the movement, the trajectory of small numbers of molecules. So it's not single molecule like I showed you before, but it's still a small number of molecules, 2, 3, 4, 5, right? So, but you have a non-direct representation of trajectory. What you have is a autocorrelation of the number of photons, and you have to infer your parameters from this. That means that you have to have a model for that, just like I showed you from Brownian motion. And currently, as I will show you, we only have 2 theoretical models from which we can write this. The 2 theoretical models for which we know how to write this is Brownian motion, that's here, and fractional Brownian motion. So that means that you can infer your parameter only if you have a molecule that behaves as Brownian motion or fractional Brownian motion. If you have the other random walk of fractal CTRW, there's at least you have no expression of G of the autocorrelation function. But you have very good temporal resolution, down to 10 microseconds. You can even be smaller if you want. And it's very easier to use and to set up in the lab if you are a non-experimental user. So that's the two main experimental approaches that you can use for that, right? Of course, as I will show you, you need to have an expression for G of tau to infer the parameters. If you don't, what do you do? You can use machine learning to infer the parameter from G of tau, even if you don't have an analytical function for it. And that's what I will show you in a moment. Okay, so the problem we want to solve now. So we have measurement, experimental measurement M, either from single molecules or microscopy or from FCS. And we want to know what is the best model T to explain M. Is it Brownian motion? Is it continuous time random walk? Is it over the form motion model that I show you? So this is a model selection. What is the best model to explain what you see? And then once you have chosen the model, what are the values of the parameters that explain what you see? So this is parameter estimation. So in fact, there are two problems. First you want to select the best model. And once you know what is the best model, you want to adjust to fit it on your data to have an estimation of your parameters, right? And sometimes it happens that for Brownian motion or fractional Brownian motion, if you have the trajectory itself, so if you have single molecule trajectories, like I show you with single molecule super resolution microscopy, sometimes for Brownian motion and fractional Brownian motion, you have the likelihood of the trajectory. So you know, in terms of Bayesian analysis, how to attach a likelihood to what you observe. I won't go into detail. For Brownian motion, it's very simple. It's the product of if you have n motion, you have n, the product of n Gaussians. It's a bit more complex for fractional Brownian motion, but still you have an experimental. So if you have trajectories with VM or FBM, you can use classical minimum likelihood estimators, whatever you want, Bayesian inference, to estimate the parameters from the trajectories. But we have no likelihood for the other moments, we have no explicitly written likelihood for the other models like CTRWE or random walk on fractals. We still wait for someone who will be strong enough to write them, but it's not the case. I'm not strong enough to do that. So what can you do? So another thing that you can do is, let's go with the MSD. As I showed you in the beginning, the Brownian motion is the only motion, the only model for which the mean squared displacement is linear with time. So what you can do is you can take your trajectory, compute the MSD for the trajectories. If it's linear with time, you say, ah, cool, it's Brownian motion. If it's non-linear, meaning if it's a power law, for instance, well, the thing you can say that it's not Brownian motion, but is it CTRW? Is it random walk on fractal? Is it, well, you don't know. So it's not that simple. On top of that, one of big problem is that, let's say that you measure your alpha and you find a value of alpha that is 0.99. You say, okay, is 0.99 close enough to 1 so that I can say that it's Brownian motion? Probably yes. If it's 0.95, so what we need is some kind of statistical test. Something that will say, okay, if it's 0.90, given my data, it's likely that it's Brownian motion. We don't have that. No one found a way to compute some kind of statistical test for that, sadly. And then it turned out that to have a correct estimation of this curve, you have to average over a huge number of trajectories, like hundreds of trajectories. And these trajectories, they have to have huge, at least several tens of time points, 30, 40, 50 time points. That's the only condition where you can have a nice average and decide whether it's linear or not linear. And usually, experimentally, you don't have that. So that's not enough. My point is that it's not enough to distinguish between Brownian and the other models. So what can you do? You can say, okay, maybe my MSD as a function of time is I will consider just one feature. It's one feature to describe my trajectory. Let's say to describe, I will describe my trajectory with other quantifiers, other features. For instance, you can say, okay, I will measure the envelope of the trajectory, measure and use it as another feature. You can say, I can measure the consecutive angles, really. I mean, when I get from A to B and B to C, what is the angle? That's the average angle or the variance of the angle. That's another possible feature. So at the end of the day, if you start to do that, what you have is for each trajectory, you can have a number of features, different features that describe this trajectory. Well, if you have features, you can run simulation with a known model. You can say, okay, I know that I will simulate trajectories with CTRW and with this value of alpha. I will compute the trajectory, the features. And if I do that, I can just do very simply machine learning like gradient boosting to try to do model selection. In this case, model selection is just classification. Your machine learning, you will train your machine learning algorithm to decide whether the trajectory is CTRW or whatever. And to do the inference, it's just regression, right? So it can work, and it's exactly what's the state of the art. What is done in the literature nowadays is that you start with that's simulation-based inference. So you said that you have, for instance, 4 models: BM, FBM, CTRW, and RW on a fractal. And then you use your computer to simulate, to produce trajectory samples that correspond to each type of model, right? Then you define your features, you extract the features for each of the trajectories, and you use a machine learning or deep learning model. It's supervised, it's a completely supervised problem, because you know exactly for each trajectory what is the model, you know exactly what is the value of the parameters. So you can train your model to do the model selection, so that it will be classification or the regression, and you can learn to know what is the model and learn to guess what are the parameters. So this, you will train your model on synthetic data, simulation-based data. And once that your model is trained, then you forget that. You take your measurement coming from single molecule trajectories, for instance. You extract your features and you feed it to the model. And so just a forward stuff, and you will see if it's what model it is and what parameters it is, right? And in fact, that's the state of the art for the single molecule trajectories. It turns out that since 2020, it's been 4 years ago, some people have launched a challenge on the internet, just like Kaggle challenge. It's a type of Kaggle channel, and it's just a channel where you have trajectories, and you have 3 tasks in the channel. You have to decide what is the model, so model classification. So they have 1, 2, 3, 4. It's a bit the same. You see CTRW, FBM. They have other models different than those that I've shown you, but in the same case, different types of models. So you have to do model classification. You have to do inference of the anomalous exponent, the alpha exponents, and so on. And so it's an open challenge. It's every 4 years, and the teams all over the world can participate to the challenge to try and propose ways to measure this thing. And you see, if you like, this was the list of the teams that participated in the challenge 4 years ago. And you see the method they use. They use to do the model classification or the inference. And you see that it's always a CNN or RNN, or some are like Bayesian inference. Other, they use classical machine learning like gradient boosting to do that. The main problem is that the trajectories, they are never the same size, right? So if you take the trajectory itself and you put it to your vector is not the same size, so it's a bit like always difficulty. So what you can do LSTM, right? That's why you have so many LSTMs there. You can use STM to get rid of this fact that your input vector is not the same, never the same. Of course, one other way to deal with input that have different sizes is to use graphic models, the GNNs. And yeah, you have this part. This was made by the French group in Pasteur Institute and his Myria team. And they do graph neural networks. In fact, what they do, they use their trajectory, they translate it to a graph, a graphic representation, and then they do GNN to do the classification and regression tasks. It was done by Jean-Baptiste Masson in Pasteur. And also all those kinds of type of studies. And yeah. In fact, what they do, most of the, it depends, but most of them try to do a change point. It's just a way to find the change point. And once you have the change point, you can you have your other network that works on the two different regimes. So the main difficulty here is to find the change points. But again, it's also a supervised problem, so it's doable. So does it work? So you have here the results of the challenge done 4 years ago. So every curve is a different participant, and this one, the orange one, is the best in class. And you have the F1 score, so that's for the classification. I'm not sure I have to explain what is a F1 score. It's okay for F1 score gives you the amount, the fraction of models that are correctly classified. And you have the MAE, so the mean average absolute error for the estimation of alpha. So the error you made when you do the experiment. So of course here, when the F1 score goes to 1, it's the best, and here when the error goes to 0, it's the best. And what you see is that if this is plotted as a function of the length of the trajectory, so the number of position in a trajectory. If you have very long trajectory, like 600, 800 points, this works like a charm, at least for some of them. You see this one, the best of one is almost, it's 0.9 something in terms of F1 score, so it really works nicely. And again, for the estimation of alpha, the error is 0.1, alpha goes from 0 to 2 in this case, so it's less than 10% error. So it's not bad at all. And if you look at the MSD, here it's a curve of the same result that you have if you just measure the MSD, and if it's proportional to time. And you see that their methods are improved quite a lot compared to the MSD. So that's nice. Everybody was happy, except that one issue is that for this to work, you need to have long trajectories, like 200 or 200 length trajectory. You almost never have 200 trajectory that have 200 positions in biological experiments. In the nucleus, for instance, I've worked quite a lot with the nucleus. What you can have are trajectories that are 10 to 20 position lengths. So 10 to 20 will be like here. So your F1 score is ridiculous and your error is very large. So this means that what currently the state of the art is that we know it's very easy to do the challenge for long trajectories, unrealistic long, I will say. The challenge is still to be able to do the task when you have short trajectories. And maybe we will have to wait a bit till people find ways to solve this problem for short trajectories. So if you have one short trajectories, it's no use to try to use single molecule trajectories. I mean, it's nice because you see your single molecule trajectory, you're happy, but you will never be able to solve the problem. So if you are not able to do that, you can still try to go to FCS, fluorescence correlation spectroscopy. Yeah, that's the possibility, because of course once you have your autocorrelation, so again, you remember you compute, you record the number of photons as a function of time. You do the autocorrelation, it decreases, and the time where it decayed by a factor of 2 gives you the coefficient, diffusion coefficient, for instance. But this works for Brownian motion, and this also works for FBM. For FBM, we also have an analytical expression of the autocorrelation function. It's the same as for Brownian motion, except that you add a power of alpha here. But it's the same kind of stuff. So you can see it. But for CTRW, we have no analytical expression. For diffusion in fractal media, neither. Okay, so not a big deal. Since it's purely supervised problem, you can even if you don't know the expression of G of 2, you can do machine learning. So you can generate trajectories with BM, FBM, simulate how many photons they will emit if they are simulated, simulate the corresponding autocorrelation function, G of tau. You won't be able to fit it because you have no function to fit it, but you can take it, pass it to a classifier or regressor, and try to do model selection, so classification and inference. And it works very nicely. We did that with my student, Nathan Kiblier. Of course, we had like a bit of a big training set, like more than 3 million trajectories for 1, 2, 3, 4, 4 models. And we did the classification and regression problem with the histogram gradient boosting. We just took the hist gradient boosting from Scikit-Lab without even doing hyperparameter evaluation, optimization, and it worked really like a charm. I will show you in a moment. We did it also with deep learning. We did it with convolutional neural networks, but in fact it was not better. So we published the version with histogram gradient boosting. And this is the result that we have. So the same. So here we can have very short trajectory. We don't care because we are not limited by the length of the trajectory. So this is the F1 score as a function of the observation time. Now, because you record of course, the number of photons, if your recording time is very short, you won't have much photons, so you won't be very precise. So of course, your classification, your results, they don't depend on the trajectory length, because you have no more trajectory length, but it depends on the observation time. But even with short observation time, like 500 milliseconds, you see that we have very good F1 scores like that. And what is nice is that we have a way to have the fact that the classification is as good whatever the size of your illumination, because depending on the laboratory, and you will have different, and the microscope, this parameter will be different. And we have a way to have very good results in terms of classification without whatever the size of the microscope illumination. Now, so remember, so we have between 0.8 and 0.9 in terms of F1 score. Remember that in the ND challenge that I showed you for the trajectories, the F1 score were around 0.6 to 0.9, which is quite similar to what we have. So we are roughly as good or as bad as they are. Now, in terms of parameter inference, so I showed you the inference of alpha for the 4 different models. And you see that we are better for some models, we are better, some better, we are less precise, right? But together, we are between 0.25 and 0.2 in terms of MAE, which is much better or the same order of magnitude than what they had with the trajectory in the MESL. So that means that with this type of analysis, we are able to select and do model selection, say what is the model that is underneath what you see, and estimate the parameters of the motion. Of course, so this was based on synthetic data. We did this also on our experimental data and so on to check that it was not completely silly, and it works. I won't go into detail at that. You can read the paper if you want. Okay, and also, of course, it's always the case. The challenge there is that you can, and that's an important, because that's my former biologist part who's talking there. It's very nice to have like machine learning algorithm and to publish paper to say, okay, they work on synthetic data. And then you say to biologists, yeah, take it and use them. They will never use them. So you have to be extra careful to put them a way to use them easily, right? And that's what we did. We have an open source, we have a Python notebook that I hope it can be used by biologists so that they can just either they can use our trained model, just input their data and do the model inference and the model selection and parameter inference, or they can even retrain a model with their specific parameters and do it better. Okay, just yeah.
Your experiments simulated a 1,000 trajectories.
3 million. Is
it cheap?
Yeah. Yeah, 3 million trajectory. One trajectory is like 0.1 seconds, something like that. Plus, it's massively parallel, so you put it. There's no dependencies between the trajectories, so you do that in multi-core. It's very easy. It takes, I mean, it's big files, of course, like always, but it's not very computer intensive. We'll skip that, and maybe to go to the conclusion. So what I've shown you is that if you couple numerical simulation with machine learning or deep learning, you can do model selection in FCS almost as easily as in single molecule trajectories. That's nice. In particular, you can do that with Brownian motion, FBN, and CTRW. Oh. Yeah, it was almost the last one. So it's still here, no? Ah. Ouais. Lucky. Non, si vous l'avez là, il l'a, ouais. Ouais. Oh, it's okay. Okay, so thank you. So now you can say, okay, now you have a way, you have methods to measure the motion of the type of motion for molecules in the cell. Show me application to experimental data. I'm not a biologist, so I don't have that yet. But I think that's I mean, probably this will come in the decades or the years to come. But my point was there that, in fact, as you've seen, if you take the history, the first, so the Einstein's paper who could find an equation and find a model for diffusion was 1905. Montrol and Weiss paper for CTRW was 1965. The random walk of fractal was 80 years before Einstein. That means that if Einstein has asked the question, what if I want to do the theory for another model, then he will have to wait for 80 years. Well, 80 years is long. It's the same like what Boltzmann did in 19 or something, and it took 1 century to show that the distribution of the Boltzmann, the Boltzmann distribution goes indeed to that asymptotically. So if you wait for theoretician, it's good to have theoretician, but if you wait for them, you can wait for long times. The idea is that here, if you just do machine learning, can be compared between 2 experimental conditions, and that's what you can have here. With that, I will just. Yeah, but. Oh, it's not a big deal. C'est bon? I just have a single, one more slide. Yeah. Just if you want further reading about that. 1, 2, 3, 4 reviews on the topics. One on a theoretical aspect, a very nice review by German colleagues, Fling and Franosch, of how do you model anomalous diffusion on the biology, about how to characterize the diffusion on the Andy challenge, a very recent archive paper on how do you do the model inference and so on trajectory data. This one is very nice as well by Johan Elf about what can you learn from single molecule molecules in living cells, and this is more about molecular crowding if you are interested in that, right? And with that, I think that I'm finished. Well, so the last slide was to thank the funding. So you don't, it's not a problem if you don't see the ANR and INRIA who funded this work. So with this, I can take questions. I will be more than happy to answer your question if you have any. Thank you very much. So do you have questions? Any question? There's no stupid question. I mean, that's a sentence that you've heard many times, but it's true. Yeah.
Thank you. Maybe I missed the answer from your talk, but what is the challenge? What can you learn about a particular population of cells when you know which model best fits it?
Oh, okay. Once you know for one cell,
one given population of.
In fact, depending on the motion model, this will tell you how the molecules in the cell they move, and if you know how they move, you can say, okay, what is the probability that they encounter? So in fact, it's all the biochemical kinetics that will.
So yes, so my question was, what influences cell dynamics, kinetics?
This we don't know. No. But for instance, what you know, for instance, if you have anomalous diffusion like CTRW, that means that your mean square displacement, your workers, they mostly stay at the same place. They keep.
Is it about cell interactions? Le frottement, comment est-ce qu'on dit?
No, yeah, we don't.
Cell tribology.
Yeah. No, we don't know yet. Really, it's for sure. This is supposed to change biochemical kinetics. That's for sure. But how does it change it? We'll see much more. Thank you.
You have shown during the second part that your research is more and more machine learning oriented. So my question is, if you are looking for an intern or a PhD student, what kind of profile actually you are looking for? Someone who is more biologist or computer scientist or AI expert? So.
Yeah, first, I'm not sure I have such a choice, first of all. No, it's very difficult. I mean, biologists usually up to now, it's very difficult to have biologists who have the expertise in modeling and in machine learning to do this kind of stuff. So usually, either we're working with physicists by training, or computer scientists, or mathematicians. Usually, it's like statisticians. So it's usually the one of the three main sources of the foundation of the.
Ouais, ça me fait du bien.
Thank you for the presentation. In the conclusion, you mentioned the detection of anomalous diffusion. And so I was wondering what's the challenges there, because I guess that you don't have any data that are anomalous in some sense. So you're just trying to compare with simulations, I don't know, things like that. So what are the challenges? How could you detect these kind of anomalous behaviors, and how do you make use of the simulations to try to detect this?
Yeah, sure. That's my fault. I wasn't clear on that. What is called anomalous diffusion is any thing that is not Brownian. So as long as your mean square displacement is not linear with time, by definition, is what people in the domain call anomalous diffusion. So it's not really anomalous in the sense that it's not normal. It's not, people should use non-Brownian instead of anomalous, but it's the vocabulary of the field. Sorry?
The detection of
Yeah.
It's relatively easy, or you need lots of data to.
It's the same. I mean, when I show you the F1 score, it was model selection, and one model was Brownian, and the 3 or 4 others were anomalous. So when you do that, you do model selection, so you distinguish Brownian from anomalous. And it's easy. Of course, it's easy if you have large, long trajectories. But again, it's more difficult if you have short ones. It's always the same issue. But again, it's a question of statistical estimation. If you want to estimate the distribution of a statistical variable, if you have you sample your distribution, if you have just a small number of samples, it's very difficult to infer the distribution. If you have a large number of samples, it's much easier. I mean, it's just a p-value in a way. Yeah.
Yeah, thank you. So I am a biologist, so sorry with the mathematical model. I got a little lost, but we know that the trafficking of the molecules inside a cell is closely related to the organization of the cytoskeleton, which is itself highly dynamic. Is there a way to link these two things?
Oh, yeah.
Yeah.
I mean, yeah, it should be, because in fact, with this kind of measurements, it's something that you can do in living cells. So you can try to synchronize your measurements with, for instance, I don't know, the cell cycle, for instance. You could try to do the measurement when you perturb the cytoskeleton with all those molecules that perturb. This is something that you can try to do. It has not been done in the literature, I'm not sure. Because in fact, this kind of question, up to now, they were rather approached by physicists, and they were more interested in the model and the equation, like I did, than what happens if, in terms of biology, for instance, what happens if my cell is spreading on fibronectin or ECM stuff, or what happens if on glass? Is it different? This is a very good question, but it should be possible to tackle them with that, but nobody did it. I don't think so. Yeah, probably because your cytoskeleton becomes some, at least depends on where you look, but close to the close to the extension. I mean, at least if you're coated on the surface, you start to have like maze-like structures, and those maze-like structures, they can give you either some kind of fractional Brownian motion because it's a polymer that vibrates and your molecules it gets collides with the polymer that vibrates, or because you have to sneak into a sort of fractal-like structure, and that question will, and that would be, I would love to see those results, but to be honest, I don't know if you, I mean, if you take classical confocal microscopy, the time duration between the first guy who said, "Oh, I have a good idea to do confocal microscopy," and the time when it became so obvious in all the cell biology labs that everybody does and uses confocal microscopy was maybe 50 years, 60, I don't know, 40 years, something like that. Here, this super resolution microscopy, this dates back from 2010. So maybe in 40 years from now, it will be very, I mean, everybody will have that in the lab, in the cell biology lab, and maybe this kind of question will be tackled at this point in time, but currently not yet.
Okay, I guess that it's time for closing this session. So thank you again, Hugues.